Hello, my name is Neelam Vaidya. I'm the CEO of ViridisChem, and I have here Dr. Niteen Vaidya, who is a technical advisor as well as the CEO of Kyrosol. Today, we are going to talk about how ViridisChem's product chemical analyzer can help you and identify the environmental and health toxicity issues in novel molecules. So let's get started with the presentation. So initially, I would like to give a very quick overview of ViridisChem and its solutions, and then we'll go into the discussions. ViridisChem was started with a very specific goal of addressing certain market needs. Costly early stage R&D efforts. We wanted to help them during drug discovery time. High risk drug targets are not identified until significant R&D efforts is put into place. ViridisChem can help you by providing the full toxicity information of the novel molecules even before you start the R&D. And it also provides you 48 physical and toxicological properties, so you understand the molecule much better. Second issue, the risky late stage changes during product development. Lack of toxicity focus during early product development forces costly late stage changes when the options are limited. Traditionally, scientists would not pay attention to the toxicity of the starting material as much, and they would go with their intuition on what the chemicals are toxic and avoid the chemicals that way. But with more than 100 million chemicals, known chemicals, this intuition is not really feasible. What we provide is a toxicity information of any chemical, whether it is known, unknown, or new molecules. So that, and we also have features that allow you to identify less toxic chemicals based on your reaction needs. You may need specific functional groups or certain properties value range, or you may want to avoid certain health issues or environmental issues. By providing all those criteria during the search, chemical analyzer provides you with the options with the number of chemicals that are less toxic, and it also provides you the toxicity profiles of those chemicals. As a result, you can, right from the beginning, you can use less toxic chemicals, and the process optimization work gets much reduced. Regulatory compliance is another issue. There are about 140 different regulatory lists worldwide that are constantly changing in recent years. They are not published in a regular manner, in a regular place, or they are formatted differently. And most of them are also very textual. So compiling that information in any manner is difficult. ViridisChem has keeps this information up to date. We have about 136 different regulatory lists that we keep track of. And as the updates happen, we update our database. We have also translated all the textual information into numeric format so that we can use that to compare chemicals and show you the toxicity issues. Availability of toxicity data for starting material is very difficult. 80% of the MSDS available are either incomplete or not very reliable. Toxicity information of only few 100,000 chemicals is available out of 100 million existing known chemicals. ViridisChem has collected information from all the different sources for about 93 million chemicals. If the chemical is not known, we actually run prediction models in the background to get the estimated data, properties data. and also provide 48 different properties per chemical. And based on these properties, we provide you the toxicity scores, ecological, health, and safety scores. So having all this information available, the scientists can make much better decisions. So our product, chemical analyzer, is used during drug discovery to understand the toxicity of new or unknown chemicals. Avoid during product development, you can avoid the toxic raw materials and minimize the toxic waste using chemical analyzer. It also provides you the regulatory implications of chemicals. If the chemical is listed under any regulatory list, it is shown there. We can also help you in lab safety. If we have access to if you connect over your chemical inventory database with our product, we can actually identify where in your lab all your fire hazards or health hazards are. Having the information on all the chemicals available on the web-enabled devices, scientists are also better prepared for any eventualities. Our product is also used during chemistry education, specifically green chemistry education. Drawing the structure, modifying the structure, and looking at the differences in toxicity of the chemical provides the students a lot more insights. So our database platform is very powerful. We have 93 million chemicals, 3.5 billion properties, data collected from more than 200 different sources. We have large amount of experimental data, and we have 189 prediction models that we can run in the background in real time to provide you information in seconds. We also cover 135 global regulatory lists, and some of the corporations have provided us the data. We are also working on some deep learning algorithms to increase the accuracy of the data, but it's not in the production yet. We use the prediction models that provide minimum 80% accuracy. For properties information, we actually have the threshold of 90% plus. So all the prediction models we use are very well reputed. And then we have proprietary scoring algorithms to measure the toxicity in environment and health and safety. Some of the features during drug discovery, you can provide the molecule, we run the prediction models, and then on top of based on the properties, we actually have our algorithms that provide you the toxicity scores. During product development, if you know a problematic chemical, you can provide the search criteria based on your reaction needs, and you get the list of alternatives and their toxicity profiles. And then you can select 3 or 4 best alternatives and try your experiment further. We can also provide you all the information needed for STS. For any given chemical, we have all the data, we provide that data, experimental data. Wherever there are data gaps, we run the prediction models and provide you that information. So as a result, you have all the properties, the ecological harmonized GHS scores, pictograms, and NFPA classification and pictogram. In future, very soon, we will be also developing a module that will actually develop the STS for you. And these are all the properties we provide. As you can see, they're pretty comprehensive. And these are all the scores that we provide. There are 58 different scores that what they call an endpoint that comprise the ecological health and safety scores, and they're based on NC 355 standard. So now I'll hand it over to Dr. Vaidya, who will discuss a specific molecule, Viagra.
Okay, here we have chosen Vigara, which is a phosphodiesterase type 5 inhibitors for these studies. And there are 3 molecules which are commercial success and is in the market, and they're all FDA approved. Vigara was introduced by Pfizer. Levitra is by Bayer, GSK, and Schering-Plough, and tadalafil or Cialis by Lilly Icos. And their respective properties are given on this toxicological diagram. And if you see the properties, as far as the physical, if you see the structure of these 3 molecules, especially Vigara and Levitra, they are very closely resembling structures. Only difference is that there is a bridgehead nitrogen in Levitra that gives you significant biological activity difference, and also it increases the half-life of the molecule and some added benefits. And Cialis is completely novel in nature, a complete novel structure. Vigara has a skin sensitizer, and Cialis has eye irritant and carcinogenicity and neurotoxicity. And Levitra has some oral sensitivity issues. All these drugs are persistence. Important score for drugs are ecological score, health score, soil score, and water score. As far as the physical properties are concerned, our database provide CAS numbers, melting point, vapor pressure, flash point, and the green section of the table describes the experimental values, and other are predicted values. We also provide SMILES, INCHI, formulas, IUPAC name, IUPAC names, and functional groups gives you specific properties of the molecule, what functionality is there in the molecule. And also we provide rule 5, Lipinski rule, which is nowadays a industry standard for identifying the drug molecule. So in all these 3 cases, it's obey the Lipinski rule. That's why it is true all 3 times. And if you see the biological properties, again, the green values are the experimental values, other values are theoretical values. It also defines some LC50s and some other properties which are very critical: log KW, BAF, log BAF, BCF, log BCF with ETC, Henry's constant, KP, KOA, log KC, all those very critical physical chemical properties. Also, it describes some LC50 values for soil as well as water solubility, especially for Vigara. You can see quite difference in solubility. Here we are describing 2 analogs, one analog rather to this Vigara, and the analog is acid anilide diphil, and the difference is sildenafil has a sulfonyl moiety there. Acidanafil has a ethyl carbonyl moiety, and you can see substantial difference in the properties. In terms of sildenafil shows high skin sensitivity, oral toxicity for acetanilide is there. Sildenafil shows high ecological score, soil and health score is also very high. But all this analog also is not FDA approved, other than the first 3 which we mentioned. All these are sort of used as herbal products, and they are having some aphrodisiac effect, and they are all very easily available through nutritional product companies. And there some of the physical properties are mentioned, toxicological properties also mentioned on that chart. And there are other 2 molecules, other 3 molecules are there, which describes the eldenafil, thioeldenafil, nitrosoprodanafil. And if you see the structure, they are all related to sildenafil, and there are especially thioeldenafil and nitrosoeldenafil are the prodrugs of eldenafil, because after the hydrolysis they produce eldenafil. And if you compare the structure, for example, the difference between thioeldenafil and eldenafil is it has this instead of carbonyl is a thiocarbonyl group there. And nitroso-protanafil has a nitroso group there, and it produces nitric oxide, which is also critical for biological activity. And sildenafil shows high sensitivity again, high skin sensitivity. Nitrophosphoranafil show high genotoxicity. Sildenafil shows high flammability and also has some long-term effect. And thioildenafil shows high toxicity and oral toxicity, and all shows high persistency. That's it, yeah.
Thank you very much. I hope you enjoyed the presentation. Please feel free to try our product just by going to ViridisChem.com, and there is a button for free trial. I hope you like the product. Please give us the feedback. Thank you.
Read the last slide.